Hello viewers, this is Hemalata, lecturer in Zoology, Government Degree College, Permanent, Kurnool. Today in this class, I am going to tell you about DNA fingerprinting. The learning objectives of my topic are: the student will be able to describe the process of DNA fingerprinting and to learn which part of the DNA genome is used for DNA fingerprinting. Understand the factors that can increase the accuracy of DNA fingerprinting. To interpret DNA fingerprints. To understand how DNA fingerprinting can be used in the chemical investigations. And to explore various uses of DNA fingerprinting. Now let us see what is DNA fingerprinting. DNA fingerprinting is also known as DNA profiling or DNA typing or genetic fingerprinting, genotyping, or the identity testing. It was developed by Alec Jeffreys in 1984. He is a geneticist. He is known as the father of DNA fingerprinting. In the Indian prospect, Dr. Lalji Singh is the father of DNA fingerprinting. He has solved Rajiv Gandhi's murder case. DNA fingerprint is an analysis of nitrogenous base sequence in the DNA of an individual. Now let us see the background of DNA fingerprinting. Almost every cell in our body contains our DNA. On average, about 99% of the DNA between two human beings is the same. The remaining 0.1% is what makes us unique, unless the identical twins, they have the same DNA pattern. Although this might sound like a small amount, it means that there are about 3 million base pairs that are different between two individuals. These differences can be used to help distinguish you from the other persons. Now we have to know about the genetic markers in the personal identity. DNA of an individual carries some specific sequences of bases which do not carry any information for the protein synthesis. Such nucleotide sequences which do not carry any protein synthesis information are repeated many times and they are found in many places throughout the DNA in the form of RFLPs, VNTRs, STRs and SNPs. RFLP, repetitive fragment length polymorphisms. VNTR, variable number tandem repeats, and STRs, short tandem repeats. SNPs, single nucleotide polymorphs. The number of repeats is very specific in each individual. These short tandem repeats are much shorter sequences of 3, 4 or 5 bases that are repeated many times, and these repeats are the ones which are used as genetic markers in the personal identity. So we know that every individual is unique here. By seeing this slide, you can confirm it. See, in this slide, 3 individuals A, B, C. In their genome, there are some base pairs which are arranged repetitively. These are very short, a sequence AGAT, AGAT, AGAT. These AGAT is common, but the number of repeats in the site is differing in each individual. For example, in the site 1, there are 7 repeats of AGAT in the individual A, whereas AGAT is in 3 repeats in individual B. In the C, there are 5 repeats of AGAT in the site 1. Whereas in the site 2, the AGAT sequence is again repeated, but it is present in 6 repeats. Whereas in a B individual 8 repeats, in a C there are 11 repeats. So these short tandem repeats are the one which are used for DNA fingerprinting. Let us now see the protocol of the DNA fingerprinting. First, we have to obtain the sample cells for the DNA. Later, the DNA should be isolated from these cells. The next step is fragmenting the DNA. The next, separation of the DNA fragments on the gel electrophoresis. The next step is denaturing the DNA into the single-stranded nuclei, single strands. And the later, the blotting technique should be done. The blotting used is the southern blotting technique. Whereas the next step is the hybridization with the radio-labeled probes. And the last one is autoradiography for the analysis and the interpretation of the DNA fingerprint. So let us know the steps in detail. The first step is the obtaining the sample cells for the DNA. So the cells for the sample are collected from the human material like blood or the blood droplets or the hair roots, skin scraping or the mouthwash or the swab from the cheek or even the semen collected in the case of the rapes. So these form the sample cells. The next step is you have to isolate the DNA from the collected sample cells. The DNA is extracted in its pure form from the sample cells. So the next is the DNA should be fragmented into number of pieces by the restriction endonuclease activity. The used restriction endonucleases, they cut the DNA into numerous fragments. Later, this sample is filled in the well created in the agarose gel and the electrophoresis is run so that the fragments of a DNA, they get separated in the form of bands. The band pattern here shows the shorter fragments towards the positive pole and the longer and large sized particles at the top. The band pattern is based upon their molecular size. So as the gel which is used here is agarose, it is not a stable one, it is a fragile one. So the band pattern which is present on this fragile agarose gel is to be transferred onto the stable membrane. Before going to that, you have to separate the double-stranded DNA into single-stranded molecules. So we have to denature the DNA. The DNA on the gel should be denatured with an alkaline solution such as NaOH to produce the single-stranded DNA molecules. The next step is blotting. The blotting technique here shows we are blotting with the nitrocellulose paper. The band pattern which was present on the agarose gel is to be transferred onto the nitrocellulose filter paper or the nylon paper. This technique is called as southern blotting technique. So as a result of a southern blotting technique, all the bands which are present on the gel, they get transferred onto the nitrocellulose paper. This is the one where it is done by baking at 80 degrees centigrade. By baking at 80 degrees centigrade, the DNA strands which were present on the agarose gel, they firmly get fixed onto the nitrocellulose paper. Now we have to prepare radio-labeled probes for the hybridization. A special radio-labeled DNA probes are prepared, and you know that a probe is a single-stranded DNA molecule, a small single-stranded DNA molecule that is complementary to the gene of interest in our sample study. And at the same time, the probe is the one, the sequence of which we know. These probes, they contain the repeated sequences of bases complementary to those of the STRs. So now when the radio-labeled probes are added and incubated, these radio-labeled probes, they get attached to the single-stranded molecules which were present on the nitrocellulose filter paper. As a result, all the single-stranded molecules, they again transform into the double-stranded molecules. The excess probes, excess radio-labeled probes which are present here, they are washed off. They are washed off by using wash solution. The next is the last step, the analysis and interpretation of the DNA fingerprinting. The process of detecting hybridized band pattern using X-ray film is called autoradiography. An X-ray film is placed next to the membrane to detect the hybridized radio-labeled probes, radioactive probes. And these places, the hybridized portions, and these places, they are marked as dark bands when the X-ray film is exposed. So the last step is the X-ray film is developed. The X-ray film is developed, and this X-ray film will be showing us some bands which are similar to the barcode which we get on the bill paper. So the X-ray film is developed to make visible the pattern of bands which is known as a DNA fingerprint. This is our identity card or the individual barcode. Now let us see the applications of DNA fingerprinting. As it is a chemical test that is showing the genetic makeup of a person or the other living organisms, it is forming the biometric security. It is involved in the protection of the endangered species by maintaining their records for identification of tissues and of the dead endangered organisms. Means it is used in the conservation of the wildlife. It is having the taxonomical applications in the study of the phylogeny. It is also having the applications in the pedigree analysis. In a pedigree analysis, the inheritance pattern of a gene through generations is studied. It is involved in the anthropological studies. So, how the humans have been evolved from the apes. Means the charting of the origin and the migration of the human populations. It is having the applications in the medico-legal cases. In medico-legal cases, clearly you will be establishing the paternity or the maternity more accurately. To solve the paternity and maternity issues, the DNA fingerprint is the best one. And it is also involved in the forensic analysis to detect the crime, and you have to confirm the suspect in the crime. So the positive identification of the suspect is done by using this DNA fingerprinting. So now I think that you have understood the topics. Let us solve one problem. Who is the criminal? Let's solve it. So here is a band pattern which is showing the DNA fingerprints of different persons. The sample which is obtained from the crime scene, from the victim, and 3 suspects 1, 2, 3 are here given on the agarose gel slab. From this agarose gel slab, let us confirm who is the culprit. So the band pattern in the crime scene should match with any one of these 3 suspects. Let us see. The last short fragment is here matching with the suspect 2. This band is matching with again the suspect 2. This band is again matching with this. Again this is matching with suspect 2. Again the crime scene band here, it is matching with suspect 2. Again it is having the match with here. This again having a match with the suspect 2. So all the band patterns which are present in the crime scene region, they are matching with that of the suspect 2. So you can confirm that the suspect 2 is the culprit. He is the criminal. He was involved in the crime scene. So by this you have understood the DNA fingerprinting. What is the protocol followed in a DNA fingerprinting, the applications, and how to solve the criminal cases. Hope you have understood the topic. Thank you for watching.